All right, well, good afternoon, everyone, and welcome to the company presentation portion of the Regenerative Medicine Investor Day program. I'm Robert Uhl with Westwick Partners, and I'm based in San Diego. And it's my pleasure to introduce our first company presentation this afternoon, which is also based in San Diego, Fate Therapeutics. So with that, I'd like to turn it over to the President and CEO of Fate, Christian Weyer. So take it away, Christian.
Thank you, and good afternoon, everyone. Let me kick off by reminding you that my presentation will contain forward-looking statements that are subject to risk and uncertainties, and refer you to our recent filings with the SEC for more detailed descriptions of those risks. Fate Therapeutics was founded in 2007 and ever since has worked to pioneer a very novel approach for intervention in stem cell biology, that is the pharmacologic modulation and programming of adult stem cells. We have applied these methods and novel approaches to generate a therapeutic pipeline that we are applying to the treatment of rare, life-threatening disorders. With one of our approaches, the ex vivo modulation of hematopoietic stem cells, we have already established clinical proof of concept, actually for hematopoietic stem cells as well as T cells, by studying or completing a clinical study in patients with hematologic malignancy who are undergoing hematopoietic stem cell transplant. Building on this clinical proof of concept, we have advanced our lead product candidate, ProHema, into phase 2 development. And looking beyond phase 2, we believe there's an opportunity for a very capital-efficient path to an initial BLA. Beyond hematologic malignancy in the transplant setting, we have developed a pipeline that is poised for clinical data readout in a range of rare genetic disorders, including lysosomal storage disorders and muscular dystrophies. We have assembled a very strong group of scientific founders that are experts in stem cell biology, an experienced management team, a foundational IP estate, and with our IPO last fall, we have the cash position to advance our pipeline to key value inflection points. So I think the key theme of this morning was that I think that there's a growing sentiment that the field of regenerative medicine and stem cell science really has the potential in the next 5 to 10 years to dramatically change the face in many areas of clinical medicine. I'm just going to highlight 3 areas where we believe and have strong conviction that some of this progress will actually be made. First off, with respect to the use of cellular therapies for application to the patients, we believe that there is a tremendous opportunity to use ex vivo modulation to optimize and tailor these various cellular therapies for patient use, improve patient outcomes. Secondly, we believe it will be possible, using protein therapeutics as an example, to activate and stimulate cells inside the body as natural drivers of tissue regeneration. I'm going to tell you about one program that we are working on, which is focused on muscle regeneration using a Wnt protein. And lastly, we believe it will be possible in the next 5 to 10 years to see dramatic advances based on the induced pluripotent stem cell technology. This was recognized, I think, this potential in the 2012 Nobel Prize. Initial clinical trials are starting, and we have spent the last several, 5-6 years to develop a industrialized platform that we believe positions us well to enable the therapeutic application of this exciting technology. Let me go into a little bit more detail with our first program and franchise, the Hema Franchise, which is based on the ex vivo modulation of cells. What you see depicted here is the overall therapeutic paradigm, which is quite straightforward and simple, in that we can take an unmodulated cell, whether that is a hematopoietic stem cell from various sources, isolated hematopoietic stem cells or T cells, expose them to pharmacologic modulators, and within a matter of hours, substantially improve or change their functional properties just before these cells are administered to the patient. As an example, what is actually shown here is a confocal microscopy of a hematopoietic stem cell that is with a green stippling indicating cell surface expression of CXCR4. We are dramatically upregulating, as one example, the expression of CXCR4 by exposing these hematopoietic stem cells to prostaglandin E2, and this discovery was subject of a Nature paper in 2007. By doing so, we intend to optimize the properties of these cells once they're administered to either animal models or the patient ultimately. In preclinical models, this exposure of a hematopoietic stem cell, in this case cord blood derived, to prostaglandin E2 for just 2 hours led to a substantial several-fold increase in the ability of these cells to home to the bone marrow niche and start the engraftment process, into a several-fold increase in the ability of these cells to home to the central nervous system, engraft there as microglial cells, and become cellular source of enzyme replacement, which is relevant for certain lysosomal storage disorders. We have also produced preclinical evidence that these cells with increased CXCR4 that are modulated have an increased ability to home to sites of ischemic injury and start cellular repair mechanism, as shown in the red stroke model on the bottom right. And we have also recently found evidence that T cells within a cord blood unit respond to prostaglandin E2 as a first paradigm or example of a paradigm for the pharmacologic modulation of T cells. We have quickly and successfully translated this into clinical proof of concept, working with the Dana-Farber and Mass General in adult patients who are undergoing cord blood transplant for the treatment of hematologic malignancies. With regular cord blood transplant, as you can see in the black curve, these patients oftentimes take many weeks until they engraft after the transplant procedure. And at 100 days, 1 out of 4 of these patients, unfortunately, will have died or experienced graft failure. In an initial cohort of 12 patients where we did the modulation process in one of these cord blood units, generating what we call prohema, we have shown that all of these patients were engrafted by day 30 and that none of the patients had died by day 100. This obviously is a relatively small sample, but we believe provides a strong basis upon which to develop our initial product for this indication of hematologic malignancies. This optimized cell, prohema, in this clinical study and in ongoing clinical study is actually produced on-site in close proximity to the patient and in a manner that fully leverages the existing infrastructure and the cell processing facilities of these transplant centers. So in this case, with cord blood, these cells are currently thawed, washed, and directly infused into the patient. We fit into this paradigm for 2 hours. We expose these cord blood units to the methylprostaglandin E2 modulator. That modulator is then actually washed off, and it is the cell that is infused into the patient, and it is the regulated product. It's regulated by FDA as a cell therapy under CBER. In addition to the effect on hematopoietic stem cells, we recently announced results, and this was based on a publication from our collaborators at Harvard Medical School, that there was also evidence that prostaglandin modulated the T cells in these cord blood units. There were some important and intriguing findings with respect to the functional properties, especially of naive T cells, which are critical for immune reconstitution in the first 100 days after transplant. And consistent with this finding, we observed an intriguingly low rate of viral reactivation in the initial 12 patients that were enrolled in the phase 1b study that I just shared with you. This provides a potential additional product benefit of our prohema product, and also speaks to the potential of this platform to modulate the properties of T cells. Building upon the clinical proof of concept study, we have optimized the prohema manufacturing process in that we have exchanged the previous media in which the modulation occurs with an optimized proprietary media, and have demonstrated that in preclinical models, this manufacturing change led to a further doubling of the engraftment potential of these cells above and beyond the engraftment potential that we had obtained with the modulation done under the condition that was used in the phase 1b study that I just showed you. We have taken this optimized manufacturing process now into the phase 2 study, which we just announced several weeks ago has now started patient enrollment. This is a randomized controlled multi-center study in 60 patients with interim results expected in the second half of this year and full results expected in mid-15. Looking beyond phase 2, we believe that there is an opportunity for a very capital efficient path to initial BLA, both from a clinical as well as manufacturing perspective. And I would also point out that we have already obtained orphan designation for this indication and are therefore eligible for fast track designation. Before I switch to the other program, one other thing outside of hematologic malignancy that we are very excited about is the potential of this product in rare genetic disorders. We have announced plans to initiate an initial clinical study in one group of rare genetic disorders, those are lysosomal storage disorders, where the enzyme deficiency manifests primarily in the central nervous system, and so where traditional enzyme replacement therapy is of limited use. Here we have shown that building upon evidence that hematopoietic stem cells home and engraft to the CNS to provide a means of cellular enzyme replacement therapy, we have generated preclinical data to indicate, as I said earlier, that this modulation process drives more cells to the CNS, and we have measured increased expression of missing enzymes actually in rodent models. Switching to the other program, which is focused on the in vivo modulation of stem cells with an initial focus on muscle. The therapeutic paradigm here is different in that the actual therapeutic is a protein therapeutic that, when injected, has the ability to bind to a known receptor that sits on the surface of the stem cell population in skeletal muscle, which are called satellite stem cells, as well as receptors that are directly on the myofibers to drive hypertrophy, and importantly, the activation and stimulation of the division of these satellite stem cells to drive the natural muscle regenerative process. We have spent time to optimize and analog natural occurring Wnt7a, which is the actual protein that we are developing for this indication that activates this receptor. And by delipidating the molecule, we are able to make it amenable to manufacturing in traditional cell expression systems. In preclinical models of muscular dystrophy, in this case the MDX mouse model, we have seen that 3 weeks after a single intramuscular injection of this Wnt7a analog, we're seeing increased in muscle strength that was associated with a decrease in inflammation and muscle fiber necrosis. Based on these findings, we have initiated IND-enabling activities with the goal of initial clinical evaluation of this Wnt7a analog in 2015. Lastly, as I mentioned earlier in the introduction, we have systematically over the last couple of years developed an industrialized platform for the derivation and differentiation of induced pluripotent stem cells. And I think all of you are quite familiar with the potential here in that this technology allows you to take a somatic cell from either a patient or a healthy donor, program this cell into a pluripotent state, which can then actually be further differentiated into future cellular therapeutics. We are initially focusing on muscle cells and hematopoietic cells. This is the area of our expertise, but the platform itself has application in other cellular therapeutic areas as well. So with that, I'd like to close by reviewing briefly our therapeutic pipeline, which I covered. With the ProHema program, we have initiated our phase 2 study with interim results in the second half of 2014 and full results in 2015 expected. We have plans to initiate 2 pediatric studies, one in patients with hematologic malignancy as well as part of our preparedness for enablement of phase 3, as well as an initial study, as I mentioned, in pediatric patients with certain lysosomal storage disorders that would include Hurler syndrome, Krabbe's disease, and various leukodystrophies. As I said, our Wnt7a-based protein therapeutics program is currently on an IND enablement path with clinical evaluation expected in 2015. And with our iPSCs, we're actively working on preclinical proof of concept for the therapeutic application of this platform. So with that, I'd like to thank you for your attention. I think there is a breakout session where I'm happy to answer any questions you might have. Thank you.